Hi, my name is Warren Pereira Piedade. I am a PhD candidate at the biology department at the University of Kentucky. And I want to share a little bit of my data that I entitled "Photoreceptor Cell Development Requires CDHR1 Protein Stability Controlled by Siah-E3 Ubiquitin Ligase During Zebrafish Eye Development." I was supposed to present this data at ASBMB 2020. So in our lab, we're interested in understanding developmental biology, but more specifically, we want to understand the molecular machinery that drives eye development. Since we have a lot of congenital blindness disease, and most of these disorders don't have a treatment available, most people develop blindness. So we want to understand how the development of the retina happens and what are the molecular machinery. And specifically for my PhD, I wanted to take a look at post-translational modification to see how protein interaction could change the development of the retina. So post-translational modifications are a group of modifications that change protein activity, giving more complexity to the same pool of proteins. One example is the ubiquitin proteasomal system. So the system is very simple. It comprises of three enzymes: the E1 activating enzyme, the E2 conjugating enzyme, and both of these enzymes are responsible for putting the ubiquitin to the E3, and then the E3 is the key main player, which is going to give specificity to the system because it will only interact with specific substrate proteins. So then the substrate will be polyubiquitinated and degraded by the proteasome. So this system caught our attention because one component in Drosophila was identified and had a homolog in vertebrates called Siah. So the Siah-E3 ligase have a homolog in Drosophila called 7 in absentia, or SINA, simply SINA, and SINA is responsible for the development of the R7 photoreceptor cell. So then we look at the Siah, the vertebrate homolog, and we saw that the protein has two different compartments. So close to the N terminal, we have the really interesting Eugene domain, or simply ring, that associates to E2 ubiquitin ligase. But close to the C terminus, we have the substrate binding domain, which interacts specifically to the substrate by binding to a degrowth signal. So proteins that have that sequence of amino acid, the one over here, will be interacting with Siah, and Siah will lead to degradation. So the VP motif here is the most important part of the degrowth signal. And in our case, for zebrafish, they express both Siah1 and Siah2L, and they are both responsive to the same degrowth signal, which indicates that they might have be responsible for degrading the same pool of proteins. So then we look at the zebrafish proteome, searching for proteins that could contain that degrowth signal, and we found among 70 of them one that was important for us called CDHR1a because it was related to eye development somehow. So it was related to photoreceptor cell development, and if we look to the photoreceptor cell in the retina, we can see that retina is a multi-layer organ. We can see that it's composed of a lot of different neurons. In the most outer part of retina, we have the pigmented epithelium, and then close to the pigmented epithelium, we have the photoreceptor cells, EI rods and cones. So the rods and cones are going to be responsible for perceiving the light and then transmitting the light to the other parts of the retina to be transmitted and also to be sent to the brain. So we have in the inner nuclear layer, we have the horizontal, the milliglial bipolar and amacrine cells, and then to the retinal ganglion cells, we have them in the retinal ganglion cell layer. So the signal will come from the light perceived by the photoreceptor cells and then transmitted through these cells to the brain. So if we look at the CDHR1 is a cadherin. It is expressed specifically in the outer nuclear layer where the photoreceptor cells are. So here, by using homeostasis hybridization, we can see the expression being localized to the outer nuclear layer. And in humans, it has been already characterized a lot of mutations in CDHR1 that leads to cone rod dystrophy. So it's a very important protein. And what we believe is that CDHR1 might be responsible for regulating photoreceptor progenitor survival and also cell development. So if we look at the photoreceptor cell, we can see that it's a highly differentiated cell, and it's divided in two segments. The inner segments have the nuclei and also all of the other cell organelles, but then in the outer segment, we have discs that are making in an octopile shape, and all of these discs contain proteins that are responsible for being excited by the light. So you can see here that in this region in between, we have two vesicles that aren't surrounded by membranes, and this region is called the connective cilium. So a lot of papers have described CDHR1a to be localizing this region of the connective cilium, and then Burgoyne recently described the CDHR1 is actually binding the immature vesicles, the one that weren't surrounded by the membrane, to the cell body. So what happens is, since these vesicles get exposed to light, they get damaged throughout time. So the cell of the photoreceptor needs to create new vesicles, and then these vesicles get internalized in the outer segment, and then the vesicles from the top get degraded by the retina pigmented epithelia because they're already too damaged. So it's a continued movement or renewal of these discs. So what we believe is that CDHR1 might play a role in the regulation of this renewal of these discs. But to understand a little better, Retina and collaborators created a mouse that it was a knockout for CDHR1, and we can see that already in the first month, it has a decrease in the number of cells on the outer nuclear layer, and it progresses to degeneration by 6 months. So what we believe is that Siah is responsible for regulating CDHR1a in order to control photoreceptor renewal, differentiation, survival, and regeneration. So in order to do that, first, we characterize its gene expression, co-localizing by Siah. And we can see here by using fluorescence in situ hybridization, we can see Siah in the outermost part of the nuclear layer and co-localizing with CDHR1. CDHR1 is very specific to the outer nuclear layer, but Siah is also expressed in other parts of the retina. When we look at Siah2, we can see that it has basically the same pattern. Siah2 being expressed around the eye into several different layers and co-localizing with CDHR1 in the outer nuclear layer where the photoreceptors are. Since post-transition modifications are protein-protein interactions, we wanted to look to see if we can characterize and mark these proteins into specific photoreceptor cells. So we take advantage of one of our transgenic lines that express GFP in the rods, so the XOPS GFP, and we can see here in green are the rods from 4 to 6 days, and we can see in red the immuno-histochemistry for CDHR1 in these sections. We can see that CDHR1 co-localizes in the connecting cilium of the rods. When we use another transgenic line to mark the cones, we can see again, we found CDHR1 being localizing to the connecting cilium of the cones. So we wanted to take a look and see how Siah was localizing. And as you can see here, by using the XOPS GFP line marking the rods, we can see that Siah, again, corroborating our in situ data, it is highly expressed in other parts of the retina as well, not only in the outer nuclear layer, but it does localize to the connecting cilia of the rods. And when we look at the cones, we got the same thing. So it does co-localize to the connecting cilia of the cones where CDHR1 was found. So now we know that Siah and CDHR1 co-localize in the same region of the connecting cilium. So we wanted to know if Siah can regulate CDHR1 protein stability. So we use a HEK cells to express CDHR1 by itself, and we get the band here by doing Western blot. When we co-transfect the cells with CDHR1 and Siah, we can see that the CDHR1 band completely disappears and we get the Siah band here. But when we use a dominant negative version of Siah, we got the rescue of the Siah. And if we inhibit the proteasome activity by doing MG132 treatments, we also get the band, the CDHR1 band back. So with this data, we can see that Siah is indeed able to regulate CDHR1a protein stability, and when we inhibited the proteasome, we were able to rescue. But we wanted to take a look more closely, specifically to the Siah, when we created a CDHR1 insensitive version in which we mutated the degrader motif that Siah could target. So if you look here, We can see the wild type being expressed in the cells after the Western blot. Here, the LMA, the insensitive version. When we transfect CDHR1 with Siah, we get a decrease in the intensity of the band. When we do the LMA, we don't get that change. When we increase the concentration of Siah along with CDHR1, we get completely disappearance of the band. However, the LMA, the insensitive band, is still there. So now we can see that by mutating that specifically degron, we get the abolish of the control of Siah over CDHR1 protein stability. So finally, we wanted to prove that Siah can physically bind to CDHR1, and in order to do that, we made co-immunoprecipitations by pulling the CDHR1 through the flag tag by using anti-flag. And you can see here, when we co-transfect the cells, we pull the CDHR1. When we co-transfect the cells, CDHR1 and Siah, and pull the flag, we were able to rescue the Siah, proving that it was physically bound to. And then by using the delta wing, we were also able to bring the dominant negative with. So then our next step was, now that we know that Siah can lead CDHR1 to degradation, we wanted to know what is the consequences of modulating Siah expression through the development of the retina. So then we created two transgenic lines in which we put the Siah or the delta ring under the control of a heat shock promoter. So every time we heat shock the embryo at 38 for half an hour, we get an increase in Siah or delta ring. And we can see here, by doing homeostasis hybridization, the AB as a wild type, and the two lines, we can see that after the heat shock, the AB doesn't have that increase, but the two Siah lines have the increase. So what we designed was, we were going to do a heat shock at 48 hours, and then a second heat shock at 60 hours. That's the window that the photoreceptor cells are developing. And then we were going to collect the embryos at 72 hours. That's when the first photoreceptor cells appear. So then here, if you can see, when we heat shock the controls, we did not see any difference. But when we heat shock the Siah, we get a decrease in the number of rods in the most ventral part of the retina compared to the dominant negative and the control. When we use the cone line, we can see the same. The Siah heat shock decreasing the number of the cones. So when we wanted to look more specifically to see if it was through the proteasome system, the UPS system, and then we treated the embryos or not with MG132 or DMSO just as a vehicle, the control, and we can see that when we heat shock, there's a decrease in the number of rods in the heat shock DMSO, but the treatment with the proteasome inhibitor rescue significantly the number of rods. When we look at the cones, we can see that again, when we heat shock the Siah line, we get decrease in the number of cones, and the MG132 was able to rescue the number of cones. And again, we did a microinjections using CDHR1 messenger RNA or the CDHR1 insensitive LMA, and both injections were also able to rescue Siah's effect on the number of cones. So now we can see that it's indeed specifically to the Siah's effect. And then here again, we wanted to look at the progenitor. So here we're using for C12, which marks photoreceptor matures and immature. And we can see that here with the Siah, we have a decrease, the Siah heat shock. But then when we inject with the CDHR1 wild type and the LMA, we have a rescue of that phenotype. So when we look at every single part of the retina to see how the heat shock was affecting them, we only found differences in the photoreceptor cells. So horizontal bipolar amacrine cells and the retinal ganglion cells were the same, and we also did not see any difference in proliferations. So our next step was look at cell death, because we wanted to understand if the photoreceptor cells were not differentiating, if it was because they were dying or because there was some sort of problem in the differentiation cell. So we look at tunnel to count the number of cells, and we can see that in the Siah DMSO, there's an increase in the cell death that the MG132, the proteasome inhibitor, is able to rescue. So now we can see that the effect of Siah is probably killing the photoreceptor cells or the progenitor cells before they can actually turn into a photoreceptor cell. So lastly, we wanted to see how the function was happening, and we have a OKR system in the lab. The OKR works as a rotating disc. We put the larvae in the center. The discs has white and black spots, and then the fish follow these patterns if it's able to perceive the light. So our data shows that even after 2 days, like because we collect the samples and the 5th day, we did not see any difference, but we see a trend. So a decrease in the eye movements of the embryos that were heat shocked in the Siah. So in conclusion, what we have is that Siah CDHR1 protein co-localized in the outer nuclear layer, the zebrafish. Siah can bind to CDHR1 and lead CDHR1 to degradation, and Siah overexpression prior to the development stops photoreceptor differentiation and also increase their cell death. So I want to thank everybody in my lab, specifically my advisor, Dr. Jacob Tomolsky. I would like to thank Kayla Titi Ali E. Torres, which helped me to do a lot of the retina part of my data. I would like to thank my committee. I would like to thank Census and Fronteiras for supporting me financially here, the biology department, and the University of Kentucky. Thank you. If you have any questions, you can send me all of the questions. I would happily answer them. Thank you.